Raise your hand if you have ever had pneumonia or known someone who has had pneumonia. As you can see, pneumonia remains a major global disease burden. Klebsiella pneumonia is a bacterial organism that causes severe pneumonia, and it causes significant tissue damage in the lungs. It accounts for about 1/3 of all gram-negative bacterial infections worldwide. One of the major treatments for this infection in the past years has been the antibiotic called carbapenem. However, in the recent years, this bacteria has evolved to become increasingly resistant to carbapenem, meaning that carbapenem is no longer effective against these type of strains. It has developed over a 50% mortality rate, meaning that over half of the people who have gotten this infection have been killed. As you can see, this problem requires the investigation of other therapeutics besides carbapenem. We could test other antibiotics that are effective against similar bacterial strains. However, the same resistance we saw in carbapenem can just as easily develop due to using other antibiotics. So we took a different direction. We decided to investigate specific cells called mesenchymal stem cells because they were shown to be having a protective quality as well as having an ability to repair tissue in culture conditions. So what are mesenchymal stem cells? Mesenchymal stem cells are adult cells that go through a process called differentiation, which means that they basically become different types of cells who have specialized cell functions, such as fat cells, bone cells, and muscle cells. And these cells were shown to be effective against many different types of diseases in culture models and in animal models. We specifically decided to use mesenchymal stem cells from the lung because Klebsiella pneumonia specifically impacts tissue from the lung. And only a handful of models have used lung mesenchymal stem cells in animal models of diseases of the lung in general. So we decided to investigate the lung mesenchymal stem cells' therapeutic potential by looking at their response to what makes Klebsiella pneumonia so serious, which is its inflammation-inducing quality. So how does Klebsiella pneumonia cause inflammation? It does this through an outer molecule on its membrane called a lipopolysaccharide, otherwise known as LPS. Once Klebsiella pneumonia gets into the body, it releases these molecules, LPS, and these LPS molecules target immune cells, specifically macrophages and neutrophils. And it prompts them to rapidly release a lot of inflammatory cytokines. And this rapid release of inflammatory cytokines is what caused severe inflammation as well as tissue damage. Some stem cells have the ability to stop this production of inflammatory cytokines. So our first question was whether lung mesenchymal stem cells can affect cytokine levels caused by Klebsiella pneumonia. To answer this question, we isolated two types of immune cells, macrophages and neutrophils, and we cultured them for about a week. And as a reminder, these two cells are the cells that produce inflammatory cytokines that cause such severe tissue damage. And so we added LPS from Klebsiella pneumonia, and then as expected, we saw that they produced a dramatic level of inflammatory cytokines, eventually causing cell death in both macrophages and in neutrophils. For half of the samples, we added lung mesenchymal stem cells to the immune cells to see what was their effect on the inflammatory cytokines that were produced. And what we saw was that these lung mesenchymal stem cells were able to significantly decrease inflammatory cytokine production in both macrophages and in neutrophils. And as you can see by the red arrows, you can see that this decrease happened with many concentrations of LPS that were added to the immune cells. And what we also noticed was that these lung mesenchymal stem cells were less physically harmed by LPS compared to immune cells. And you can see that by looking at their significantly higher cell survival rate, which was 96%, compared to those of macrophages and neutrophils, which were under 30%. So from this investigation, we determined that lung mesenchymal stem cells have the ability to significantly reduce inflammatory cytokine production that causes tissue damage in the lungs. And we also saw that these lung mesenchymal stem cells were shown to withstand LPS attack physically by looking at their significantly higher survival rates. So now we know that lung mesenchymal stem cells have the ability to have a significantly higher survival rate when attacked by LPS. And so we wanted to dig a little bit deeper, wondering what causes this high survival rate. What we knew was that LPS can damage energy production in macrophages, which is what causes many of them to die. So we were wondering whether there was a unique quality in lung mesenchymal stem cells, specifically in their energy production, which causes them to have a high survival rate. So our first question was, how does LPS affect energy production in lung mesenchymal stem cells? To answer this question, we had two types of wells, control and experimental. In the control well, we just added lung mesenchymal stem cells by themselves. And in the experimental well, we added lung mesenchymal stem cells with LPS. And we compared this response to macrophages. So in the control well, we added just macrophages. And then in the experimental well, we added macrophages with the LPS. And what we measured in these 4 wells was the energy production. And we did this by measuring ATP levels, which are molecules that store energy. And the 3 things we measured were total ATP, ATP from glycolysis, which is the first step of energy production, and ATP from the mitochondria, which is where most of the energy is produced. And what we saw was that when LPS was added to macrophages, we saw that the total ATP production significantly decreased with 90 minutes of incubation. And then with 4 hours of incubation, this decrease continued and intensified. And so we were wondering whether this decrease in total ATP production was due to glycolysis or was in the mitochondria. And what we saw was a similar trend in mitochondrial ATP, which means that the LPS was targeting mitochondria. We saw that there was a significant decrease in mitochondrial ATP production with 90 minutes of incubation with LPS. And we saw that this decrease continued after 4 hours of incubation with LPS. So since these trends are similar to the total ATP production in the macrophages, we can see that the LPS were specifically targeting the mitochondria to cause a decrease in energy production. And we compared this to the response of lung mesenchymal stem cells. And what we saw was similar to the macrophages after 90 minutes of incubation with LPS, where it significantly decreased total ATP production. However, after 4 hours of incubation, you can see that the gray bar reaches close to the level of the black bar, meaning that the total ATP production significantly compensated after 4 hours of incubation with LPS. And so we were wondering again whether this was in the glycolysis or in the mitochondria. And what we saw was a similar trend in mitochondrial ATP production, where after 90 minutes of incubation with LPS, the mitochondrial ATP production significantly decreased. However, after 4 hours of incubation, this mitochondrial ATP production compensated. So from these graphs, we can conclude that lung mesenchymal stem cells have the ability to energetically recover in the long term by the mitochondria. And this ability to withstand the energy depletion caused by LPS shows their adaptive quality. So the 3 broad conclusions we can make from both of the studies were that lung mesenchymal stem cells can reduce cytokine levels that in turn cause severe inflammation and tissue damage. They have a high survival rate, meaning that they can withstand an LPS attack physically. And lung mesenchymal stem cells can recover ATP production, unlike the immune cells, like macrophages. The field of stem cell research is increasingly becoming more exciting and more relevant to the world today. However, before using these stem cells as therapeutic agents, such as using them to fight bacterial diseases like Klebsiella pneumonia, it's important to evaluate their therapeutic potential and broad qualities in cell culture models and in animal models. And thus, this study was able to do just that. This study was displaying that lung mesenchymal stem cells have an anti-inflammatory quality as well as energy recovery abilities in response to the most toxic component of Klebsiella pneumonia. And from this, we determined that lung mesenchymal stem cells can be a potentially viable option to manage this increasingly dangerous bacterial disease, having the potential to save many lives in the future.